As an early career hematologist passionate about helping treat patients with rare blood diseases, Dr. Leonard Zon set his sights on figuring out what determines blood cell fate, or the biological signals that decide a cell's purpose, and in his case, the decisions that lead to the development of blood cells. The most obvious starting place for his investigation was quite literally the beginning of life. In the inner workings of the embryo, a multicellular organism charged with the lightning fast construction of all the complex organ infrastructure it will require to survive. So he began, first with the mouse and then with the frog, but neither proved right for the job. The mouse produced too few embryos at a time to conduct meaningful scientific inquiry quickly, and the frog had other impediments. And then came an idea, a 1.5-inch tropical fish called a zebrafish, relatively new to researchers, might just be the perfect model for discoveries about human disease. Biologically, it had a lot in common with humans. Females could birth 300 transparent embryos a week, and fertilization, and thus embryogenesis and development, happened in vitro, outside the fish's body, giving researchers the ability to literally observe the origins of life, blood, and disease under a microscope. That was the hope in 1993, the year Dr. Zahn opened his first zebrafish lab. Since then, more than 30 years later, Dr. Zahn's bet has paid off. Today, he's a world renowned hematologist and stem cell scientist taking care of babies and children with rare blood diseases, from sickle cell anemia to leukemia. He's also the proud owner of the world's biggest zebrafish aquarium, conducting research on about 300,000 zebrafish in his Boston Children's Hospital laboratory. That research has resulted in foundational insights about blood development, including in pregnancy, the identification of new blood diseases, new human genes never before linked with blood diseases, a transformative stem cell related therapy for babies and kids with rare diseases, and a pair of treatments in the clinical pipeline for childhood cancer and spinal muscular atrophy affecting kids. His office walls are adorned with framed cover art from scientific journals in which he featured photos of him with influential people, many fish memorabilia, and numerous awards and plaques. And just a few days after this episode first airs, another accolade will join the existing honors in his space. That will be the March of Dimes Richard B. Johnson Jr. MD Prize, which Dr. Zahn won this year as the 2026 recipient. Dr. Zahn is one of those well-known zebrafish researchers in the world, and a doctor whose basic science has resulted in exceptional translatability, greatly improving understanding of iron transport in pregnancy for the prevention and treatment of prenatal anemia, and saving the lives of some of those vulnerable babies and children through a compound that greatly improved the success of stem cell gene therapy. And he's not done yet. Dr. Zahn is in the middle of some of the most exciting research paths of his career, many surrounding the possible role of stem cells in the development of cancer. Dr. Zahn is here today to tell us all about his career, his research, and the black and white zebrafish at the center of his world. This episode is about Dr. Leonard Zon. Dr. Leonard Zon is director of the Stem Cell Research Program at Boston Children's Hospital and the Grousbeck Professor of Pediatric Medicine at Harvard Medical School. Len, welcome to the show.
Well, thank you. Very appreciative to be here, and it's great to hear that story.
And congratulations on your prize.
Well, it's really a wonderful honor, and it really goes to a lot of the people in my laboratory over the years who've done such great work, and it's nice to have the zebrafish be recognized.
Yeah, you've definitely put the zebrafish on the map. I think that's safe to say. So I feel I know you pretty well because you've been so generous with your time helping me understand your work. And I asked to visit your lab and you said, yes, come on down. So I, about a month and a half ago, was downtown Boston at Boston Children's Hospital, and I saw your 300,000 zebrafish friends. So thanks for welcoming me into your world.
Great. Absolutely. I'm glad you enjoyed it. It's one of the top tours here at Boston Children's Hospital.
Absolutely, and you knew every single person in the hallways and in the elevators, and so, it was really cool to be, next to you that day. So, Len, your interest in blood diseases came during your time in university when you did a summer of research at Roswell Park Comprehensive Cancer Center in Buffalo, New York. Tell me how that experience led to where you are today.
Yeah, well, I went to a very small liberal arts college called Muhlenberg College in Allentown, Pennsylvania. And at the time, I really wanted to be a community pediatrician, and I was applying to medical school. But during my after my junior year, I went to Roswell Park to be able to study cancer, and I worked on antibodies at the time, which were brand new monoclonal antibodies. But I also volunteered in the leukemia ward there, and I met a woman who had leukemia, and I was very affected by that story and how she was being treated. And I always had an interest in blood cells. The blood is probably one of the easiest systems to study because you just take a tube of blood and can see all those blood cells under a microscope. So it started getting my thinking about really, do I want to just be a practicing pediatrician or do I want to have more of an academic career? And so that really was the nidus to start me on this path of being a hematologist.
I talked a little bit in the beginning about why you chose the zebrafish as your organism of choice, and one of those reasons is IVF, but not in the popular understanding where humans go to a doctor to help them have a baby, and where the eggs are removed from the woman and then joined with the sperm and then put back into the woman as an embryo. So tell me again, because I found this was one of the most fascinating things that I learned from you the day that we met. How do zebrafish mate and how does an embryo appear and develop? Because this is one of the key reasons that this is a great organism. So tell me about the very beginnings of life for the zebrafish.
Yeah, so basically, zebrafish is externally fertilized. So what happens is the females lay their eggs after a little mating dance with the males, and then the eggs are fertilized by the sperm from the males, but outside of the body. And this is actually a very efficient process. And as you say, we can have 300 embryos every week from a female developing, and so that starts the process. The sperm hits the egg, and then you see a one cell embryo become a two-cell embryo to a four-cell embryo. And eventually it makes all the organs. And it does so really by about 24 hours. Most of the organs have developed. So in humans, it takes about 19 days to get to that level. So it's a very, very fast system. And we have hundreds of embryos. The natural place where zebrafish lay their eggs is actually in the rivers, very much in shallow water. And we built a tank structure, which we called the eye spawn. And the eye spawn is a tank within a tank. And you put 100 fish in there, males and females. You pull up the inner tank, so you're in shallow water, and the fish decide to mate like mad. And we collect embryos at the bottom of this tank, and we can collect about 10,000 embryos in 1 hour. So it's really amazing. And this eye spawn tank that we build, we use all the time for drug screens and genetic screens that we're doing. So it's a very facile process.
And one of the other things that was really interesting is that they mate around seaweed, right? And the eggs, the embryos come out of the female right when the lights turn on in the laboratory, I guess to mimic the natural environment when the sun comes up.
Exactly, and unfortunately, the fish would eat their own eggs, and so we put a frit there that prevents them from getting to when the eggs drop. The fish can't get to that, but the mating dance is done around a little Easter grass in our laboratory, and so they hide and they do a little dance and then start their production of the embryos.
Let's talk about the early days, the beginning in the 90s and the early 2000s when you were cloning zebrafish genes and looking for novel ones, novel genes that could be implicated in human blood diseases. Tell me why you undertook this cloning work and talk to me about the 5 novel genes you found as a result, you and your team.
Yeah, absolutely. So, at the time, there was a concept called forward genetics. So forward genetics is you make mutant animals and then later figure out what the genes are that are mutated. And those mutations would be associated with certain diseases or phenotypes, as we say. So there was a large scale screen done at Tübingen, Germany, where a team of 12 postdoctoral fellows mated fish that had been mutagenized and looked for diseases of every organ you can possibly think of. This was done by Yanni Nusslein-Volhard's laboratory in Tübingen. A similar screen was done in Boston by Mark Fishman and Wolfgang Drever's lab. So we worked with the Germany group, and they had found mutants that didn't have circulating blood cells. And I sent a postdoc and a graduate student to her laboratory for 6 months, and they worked on all the blood mutants and then sent them back here to Children's Hospital in Boston. And so there were 50 families of fish that had mutations, and the mutations caused a blood disease. These are where mostly what's called autosomal recessive. These are where the mother and the father have a mutation. When they mate together, 25% of their offspring have this blood disease. But sometimes a mutation had happened in the same gene. And when we mated all the mutants to each other, they formed 26 what we call complementation groups. So 26 genes that it takes to make ultimately a red blood cell. And so we started to look at these really interesting diseases, and they were so fascinating, we had to go find the gene. Finding the gene was a very difficult process. We did something called chromosomal walking. So we did that process. We developed all the genomic infrastructure to be able to do this. We eventually walked through to the first gene. It took 3 years, and we found it to be a gene called ALAS2, which is the first step in heme biosynthesis. The gene was interesting only because it was the first disease model in the zebrafish. But to be fair, we already had great knowledge of ALAS2 in humans. It causes a sideroblastic anemia. So it wasn't that interesting a gene, but it did get published in Nature Genetics as the first zebrafish disease model. In our second gene that we walked through, we found something very, very novel at the time. This was a transporter, and we found that it was an iron exporter, and it was very well conserved in the fish and to humans. What happened is the yolk of the
And this is
embryo.
ferroportin, right, Len? Ferroportin.
This is a gene called ferroportin. And the yolk of the embryo has to deliver the iron in the yolk to the red blood cells, and it does it through this transporter. And the fish mutant that we had, which we called Vicerps, was anemic because it had iron poor blood, basically. And we then found in humans that this was in the placenta, and it was how iron is delivered from the mother to the baby. And then we found that only a small segment in the small intestine, the duodenum, has the expression of this transporter. And so when you eat a piece of beef and the iron from the beef needs to be delivered to your body, it does it through this gene ferroportin. And then later we found patients who are mutated in ferroportin who have an iron imbalance called hemochromatosis. And so, this was not only a very novel gene at the time, but it opened up tremendous biology that the field had been looking for years. And then we found it to be a human disease. So what happened in the original 26 complementation groups of mutants that we had is some of the genes were genes that we already knew about. But we had put them into a context and understood more about their biology. But 5 of the genes were completely novel genes. Nobody had ever really worked on them before. And then we found humans that were mutated in those genes. So by doing this large-scale screen, we had found 5 human genes that were new at the time.
Fantastic. Arguably, your biggest and most impactful contribution to science and medicine came from your work on prostaglandin E2, which you discovered is a powerful growth agent for blood stem cells. That discovery dramatically improved the success of stem cell gene therapies that saved the lives of babies and children, including those with sickle cell anemia, thalassemia, primary immunodeficiencies, and more. Now, I want to be clear that this discovery and all of your stem cell work involves induced pluripotent stem cells. All right, so what gave you the idea that prostaglandin E2 could be a special player here with stem cells? It could have this multiplying property. Um, and tell me how this lipid does this in zebrafish and in humans.
Yeah, so in the early 2000s, we developed a strong interest in stem cells. And using the zebrafish, we could actually mark the stem cells with specific genes, one gene called RUNX1 and another gene called c-MYB. And one of my postdoctoral fellows, Tristan North, discussed doing a chemical screen where you add chemicals to the water and see if you could ever find a chemical that would increase the number of blood stem cells by staining with RUNX1 and c-MYB. Now, the number of stem cells that is transplanted in a human correlates to the success of the transplant. The more you transplant, the better off the patient will be. So there's a tremendous interest in increasing the number of stem cells. So what happened was kind of one chemical at a time, we put 20 embryos in a well of a 96 well plate and added a chemical to the well and then stained that animal for their stem cells. And we did random chemical screening about 2,500 small molecules. And PGE2, prostaglandin E2, came out as the strongest hit. And it was really strong. It was very, very easy to see. Many of the embryos having, 2 to 3 times more stem cells than they normally would. So that was just so fascinating. And one of the things about the screen, which I think is very important and elegant, is you could not do this screen in any other system. Doing it
Mm.
in tissue culture would not work. It has to be done in vivo. And the fact that the chemicals can seep into the zebrafish embryos and be able to look at the region of the embryo that's making stem cells, this is a very powerful use of the zebrafish. But we wanted to see if you could use this therapeutically. And in the mouse, you can do something called a competitive transplant. Stem cells are very competitive with each other. So, what we could do is take some stem cells and outside the body treat with prostaglandin E2 and take other stem cells that are untreated. And based on mixing them together and transplanting them into animals, we could see which would work better, the untreated or the treated. So what we saw was about a 3 to 4 fold increase in the number of stem cells that engrafted with the prostaglandin. And so that was really, really huge. And so that made me wonder if we could do this clinically. And so I actually formed a company called Fate Therapeutics to execute a clinical trial on prostaglandin. And the story at the time was sometimes patients who didn't have a match donor would need another source of stem cells, maybe if they had leukemia or if a child had an immunodeficiency and they were getting a bone marrow transplant. So instead of doing bone marrow, we can use cord blood. The umbilical cord actually has good stem cells, but there's kind of few of them. So the standard of care using cord blood at the time was to give 2 cord bloods worth of stem cells. So by having this 2 babies worth of stem cells, it became possible to treat one of the baby's stem cells with prostaglandin, leave the other one untreated, and put them both into the patients. Because they're from 2 different babies, they have 2 different DNAs, and you can actually find out in their white blood cells how much came from the treated cord blood or the untreated cord blood. And what we saw was that there was many more engraftment. 10 out of 12 patients in the first trial came from the treated cord blood. So this allowed us to then proceed on and treat 150 people with prostaglandin E2. And, this works very well. It stimulates engraftment. Other people found that it also increases the transduction of viruses. And this has become a primary use of the molecule to use gene therapy. So if you're going to insert a gene to correct a child's genetic disease of the bone marrow, you can basically treat with prostaglandin, get very good engraftment, get good viral transduction, and this has helped patients. This continues, and I think that's the legacy of prostaglandin E2.
You've also got two other therapeutics in the clinical pipeline, thanks to the zebrafish. One is to treat diamond black fan anemia. That's an uncurable blood disorder affecting babies and kids. That therapeutic is approaching human clinical trials now. And then a second one is a therapeutic developed by a company founded to treat a pediatric disease called spinal muscular atrophy. I talked about that earlier, and that's currently under FDA review. Tell me about these two diseases and about these two therapeutics and how promising are they?
Yeah, so in diamond blackfin anemia, the children are born with a very low number of red blood cells. We developed a zebrafish model of diamond blackfin anemia, and then we did a chemical screen, which we call a disease suppressor screen, where we looked for a chemical that would relieve the anemia, would basically make more blood cells specifically in the disease mutant fish. And so we found a chemical increases red blood cell production. We did a clinical trial with that small molecule, but it happened at the beginning of COVID. We treated one patient. The patient did respond. And now we're in development with a company to be able to try to develop a better molecule. That particular molecule caused neurologic tics, and we're trying to engineer the tics out of the molecule and bring that into the clinic. So with respect to the spinal muscular atrophy, it was very exciting work. We published a paper in 2011 on on how the TGF beta pathway, particularly a growth factor called BMP, is able to stimulate blood cell development. And a friend of mine, Tim Springer, and I were both working on this TGF beta pathway. He had solved the crystal structure of TGF beta, and there's an integrin that physically pulls the TGF beta off the extracellular matrix. And we realized there's a way of developing drugs to TGF beta that could block the pulling off the extracellular matrix. And one of the first drugs we made in a company called Scholar Rock, we made a molecule to myostatin. So there are cows that are mutated in myostatin, which are called the Belgian blue, often known as the Schwarzenegger cow. These have huge muscles. And
Mhm.
so it is a growth factor that's a negative regulator of muscle. We developed an antibody to myostatin in an attempt to basically reproduce the cow and make humans very, very strong. So we were looking for use of our myostatin antibody and worked with the Spinal Muscular Atrophy Foundation. And they thought that our molecule was the best in class and would be something that would be really interesting to try in children who have spinal muscular atrophy. They're defective in a gene called SMN, and it's a very devastating disease. There's different degrees of severity of the disease, but many of the patients cannot walk. There are correctors for this, which actually creates a situation where you splice into an adjacent gene and kind of fix the defect because there's two genes next to each other. And this works as an incredible therapy. But still, as patients start to age, their muscle strength really goes down. So we did a phase one, phase two, and phase three clinical trial with our myostatin antibody. In the latter, we treated 180 patients with spinal muscular atrophy, and we had a substantial response. It was statistically significant, and many of the patients did very well, and some of them could actually walk with our drug. So that drug is now with the FDA, and hopefully will be approved sometime in 2026. We'll see, maybe 2027, but hopefully in 2026. And I think this will have a great impact on patients. And, you know, as a physician scientist, that's one of the most important things is to have that kind of impact. And I'm really proud of that TGF beta story and starting that company with Tim and watching this get into patients and making an impact.
Absolutely. We certainly hope that that process goes well and that you do get FDA approval this year, not 2027, and that more kids who don't have the ability to walk have that ability. So thank you for that work. Len, let's talk about stem cells and cancer. I think probably people in the stem cell community know that stem cells can be good and bad. I certainly had no idea. I thought stem cells were just all good. But when I had a conversation with you, you said, listen, we discovered that cancer begins with a stem cell state. And so let's talk about that. You found that with melanoma. Yeah, because we've known about how stem cells can help beat cancer. Actually, they can be the origin of cancer. So you found that cancer begins from one cell, a single cell that reverts back to a stem cell state. And then it becomes cancerous and the cancer grows. You found this in melanoma. Tell me about how the zebrafish helped you discover this and what is next.
Yeah, it's a really good story. So, what happened was Chuck Kaufman in the lab was studying the origin of melanoma. And, we always thought that cancer should start as a single cell, but nobody had ever actually seen this happen in the body. So, what had happened is that we have been studying melanocytes, which are the pigment cells of your skin. Now, melanocytes come from a stem cell population called the neural crest. So, during embryogenesis, this stem cell population eventually differentiates to make the melanocytes. And what we saw, he made a reporter for the neural crest stem cells. When he looked at tumors and saw that the very beginning of the tumor, he could see that this reporter came on. And it came on really early. So he decided to follow an entire cohort of fish pretty much every day for a couple months until they developed tumors. And what he could see is an adult fish, and they had one cell that was green. And 100% of the time, that one cell was the beginning of the tumor, and you could see it develop over time. So we had been the first people to ever see cancer at the one cell stage. And, we think about embryogenesis. You start as one cell and you develop. Well, the same thing is true in cancer. You start as one cell and you develop. So the surprise was the initiating event of starting a cancer was to take a melanocyte and go backwards in development. So it actually reprograms itself to become a stem cell. And at that particular point, it becomes tumorigenic and starts to grow out of control, kind of repeating what happens in embryogenesis for that stem cell population. So we've been really following this story for years, and we have a newer version of it where we see a zone of cells that are actually changed in some way, and one of the cells in the zone becomes that cell that starts the cancer. And so
Right.
now we're trying to develop therapies that would stop the cancer before it even began. And so that's going to be really exciting, and we're in the process of doing that now.
Yeah, that's fascinating. And you said that you said that you always thought that cancer would start from a single cell, right? But the
Yeah.
surprise was that it reverted back to the stem cell state. Even though that is surprising, it also makes complete sense, right? You need that blank slate in order to then morph into something new. So actually it makes sense now that we know it, of course, it makes
Yeah,
sense.
I agree. And also, over the years, a lot of people have compared cancer to embryonic tissues. And
Mm.
so, it just made so much sense once we actually saw it.
So, Len, once you get into your lab, you see all the fish, there's all the tanks, you hear the sounds. There's one guy that stands out. And I know there's multiple guys because you got a fish called Casper, and I know you got a lot of Caspers, right? Tell me, this is not a ghost, and he's not invisible, but he is transparent, which is why you called him Casper. He's got no skin pigment. Tell me, why did you make Casper, all of the Caspers in your lab, and what has Mr. Casper taught you?
Yeah, so really we use Casper fish all the time. So what happened was I had done a marrow transplant in the fish, and so we had taken stem cells from the marrow of a one fish that was marked by a fluorescent marker, and we transplanted it into a non-fluorescent animal. And I've always, as I've done bone marrow transplants on patients, I've always wanted to see the stem cells home to the bone marrow and start to engraft and see that process. But unfortunately, when I treat a patient, they're opaque, so I can't see it. So I was hoping we would be able to see it when I did this transplant, but unfortunately, the stripes of the zebrafish got in the way. so I
Oh,
couldn't see it, and it was very frustrating. So a postdoctoral fellow in my lab, Rich White, decided to take this on and find a series of mutants that could make the animal transparent. And he found two genes that, when he mutated them together, produced a completely transparent adult zebrafish. And then we could see the stem cells home into the marrow. And actually, when we make our melanomas, we can see how beautifully the tumors migrate, and we can see metastasis and other things with this. So it's a workhorse for us. We do a lot of transplantations into it. We visualize cells' movements, and also in the tumor model, look at spreading of the tumor. I think one of the funny stories, which I can tell you about, is my son was on the Olympic development team in Massachusetts, and they decided to go to Portugal. And so I'm in Portugal, and I get an email from a friend of mine, and it says, "You are famous." So I said, "Well, what did I do?" And so it turned out that we published the Casper paper, and it was picked up by the Wall Street Journal, and so we had a lot of press on a transparent animal. But it turned out that Jay Leno in the Today Show decided to do a joke about our fish. So what he says is, Red Lobster has decided to put this fish on the menu, and they said, you know, you can't see how much we've given here because it's completely invisible, and
it's
so
a
it was
really good
pretty.
deal.
It's a really good deal, exactly. So anyway, I thought that was really funny, and again, made the zebrafish famous.
Len, as we wrap up, I want to talk quickly about your research into cobal hematopoiesis, a stem cell related aging process that can lead to leukemia, including childhood leukemia. Your research on this with the zebrafish has resulted in finding out that immune cells you think that immune cells called macrophages can combat this disease process by way of, speaking of red lobster and eating Casper, by way of eating the mutant stem cells at the heart of this process. Tell me more.
Yeah, so what happens is, as we age, it's surprising that you develop specific mutations in your blood stem cells. And this may be partially responsible for aging, actually, and heart diseases and neurologic diseases that happen over time may be related to the mutated blood stem cells. So, what happens is a mutant stem cell produces mutant white blood cells. And those white blood cells are very inflammatory for some reason. They are pumping out inflammatory cytokines. And so this is an interesting process, and we've been able to study this using the zebrafish. We mutate the same version of the human genes, but in the fish, and we see what's called clonal dominance. My lab has developed a couple systems to be able to do something called cellular barcoding. We actually can barcode every cell of the body, including the blood cells. And when a mutated stem cell happens, you can see a clone start to become more dominant. So recently, what we found is that all of your stem cells are being patrolled by macrophages in a quality assurance mechanism. So what happens is, if a stem cell has developed what's called reactive oxygen species, there's oxidation happening in the stem cell. It puts a surface molecule called calreticulin. And this is an attractive signal to macrophages to get them to eat the stem cells that have these high oxidation. But there's also, in a second paper, we described a don't eat me signal called beta 2 microglobulin, and this prevents the eating. And stem cells that have activated their endogenous viruses actually leads to the production of this beta 2M, which counters this eat me signal. So you could think of it in your blood system, in the marrow, or also outside the marrow, where macrophages are constantly patrolling the stem cells, deciding whether to doom the stem cell because it has too much oxidation, or to groom the stem cell, take a little bite, and then those are the stem cells that actually divide and contribute to blood in the body. And so right now, what I'm trying to do is to teach the macrophages how to eat mutant stem cells and maybe get rid of the bad actors, and trying to make the body more healthy, particularly as we age.
That's amazing, Len. And science is so interesting, but also being able to see the interest in the science is so important, and you do that so well. So
Oh,
I
thank
hope
you.
that you can figure out how to get the macrophages to doom more of the bad guys instead of grooming them. All right, we're going to end things on a comical note, Len, because you are an enthusiastic and wonderful guy. Anyone who's ever met you knows your storytelling talents are almost as good as your scientific talents. And you already proved that because of that funny story that you told about Red Lobster and Casper. So, can you leave us with one more of your favorite zebrafish stories? From my memory, I remember 3 in particular. You could choose one of these 3 or you can do another one that I don't know about. I've got zebrafish duffel bag in European airport. I've got zebrafish transfer on Boston Turnpike, and last but not least, I've got the zebrafish resuscitation.
What happened was we were visiting Tübingen, Germany. I was visiting Tübingen, Germany, and my lab found out that I was going to Tübingen, which is a big zebrafish facility, and they decided to request some mutant lines from the Nusslein Volhard laboratory. So, I said fine, and I didn't know much about this. My lab was handling all the details, and I was visiting, and it's now the last day when I'm in Tübingen, and they tell me show up at the fish house and we'll give you the lines that your laboratory has requested. So I get there, and there is a duffel bag that is 6 feet long, and it is filled with 50 flasks with zebrafish inside the flasks, every flask having a different mutant. And I just had no idea about this scale. Now, this was before 2001, and so there wasn't as much security, but I was going to have to bring this duffel bag back with all these fish. So the first thing that happens is I go to the Stuttgart airport, and I'm thinking, I have to put these fish through the irradiator, and am I allowed to do that, or what should I do? So anyway, I put him through the irradiator, and the guard there, I feel like he's going to say he wants to look at them, and he goes, "I need to open the duffel bag." And I'm going like, "Oh my god!" So he opens up the duffel bag, and I think he's going to send me back. He pulls out one of the flasks and holds it up, and he goes, "Oh, fish!" And he goes, "Go ahead." So I get on the plane. I have this big duffel bag, and it takes up 3 compartments on the top. And so it's an hour into the flight, and I'm kind of trying to relax after going through the Stuttgart airport, and I figure I better check on the fish. So I open up the compartment, and the entire compartment is flooded.
Oh,
So three of the tanks had cracks in them, and they had partially leaked all over the place. So I rang my bell, and I said to the stewardess, I said, "Could I have some napkins?" And so she comes back with one little napkin, and I said, "No, I need a lot of napkins." So I start patting down them, and basically every hour I would pat down the water with more napkins, and it was very, very, very funny. So finally, we're flying into Boston, and they give me a card, and on the card there's a question: Are you bringing meat or fish into the country? And I'm so fried by this time, by this entire trip. I just check the box. I can't deal with it. So I now going through the customs officer, and so the guy says, "It says here that you are bringing meat into the country." And I said, "No, it's fish." And he goes, "Okay, go ahead." So we brought all the mutants back to Children's Hospital and put them in tanks, and they were all fine. Even the ones that had the cracked tanks, there was still enough water in there for them to do it. So it was a great episode in my life.
oh my goodness. So funny. So glad they all survived. That story could not happen today. Len, thank you for your levity and your incredible translational work, working on blood and blood diseases for kids and babies. Congratulations on your award. It's been so fun having you here today.
Well, thank you very, very much. Really appreciate all your energy too. Thank you.